All right, everybody. Thanks for coming tonight. Good to see some familiar faces. Just a reminder about the way that we do this. We'll talk up here for probably 30 minutes. We got a little bit of a smaller group out here. We'll probably talk a little bit longer before we open it up for a question and answer. But you all get a chance to ask a question or two if you have one. So that'll be at the end. Tonight we are lucky to have Dr. Sean Anderson here with us. Dr. Anderson is a postdoctoral research in Dr. Natalie Cohen's lab here at the Skidaway Institute. Dr. Anderson's research is focused on characterizing coastal phytoplankton communities in the South Atlantic Bight and Gulf of Mexico. He graduated with a bachelor's degree in biological sciences from Old Dominion University, a master's degree in oceanography from the University of Rhode Island, and a PhD in marine sciences from the University of Georgia. And a good fun fact is during his PhD, Sean was right here at SKIO. So he did his PhD at the Skidaway Institute, was gone for a couple years, and then
Came back, yep.
and then came back as a postdoctoral researcher. So glad to have him back, glad to have him here tonight, and glad to have you all here too. But to start off, can you just give us a general overview of phytoplankton? When we say phytoplankton, what classifies as a phytoplankton? As phytoplankton.
Yeah, sure. So when I think of phytoplankton, in my head, I'm thinking of tiny plants that live in the oceans. So these plants, though, are microscopic and they're single-celled. So similar to, right, plants and trees that are on land, these tiny microscopic organisms are doing a process called photosynthesis, where they're using sunlight and carbon dioxide from the atmosphere. They're converting that into food for themselves, so for their own growth. And as a byproduct of this, they're producing oxygen. And phytoplankton are really important. They produce half the oxygen that we breathe. And one of the differences, though, between phytoplankton and land plants or trees is that the phytoplankton populations turn over and grow really fast. So these single cells are growing on the order of hours to days, whereas a tree takes decades to centuries. But they grow really fast, so they can be very concentrated within these waters. Yeah.
Yeah. So you say waters. Where do phytoplankton live? Do they travel around or they stay put? Can you just kind of answer those questions a little bit more?
Yes. So phytoplankton, they're everywhere from coastal Georgia to the open oceans, and they're largely at the mercy of ocean currents, winds, and ocean
Mm.
circulation. So they're being drifted. So some of them can swim, but not very well. So largely they are taken by the ocean currents. Now, phytoplankton too, as we'll see. So with this image, right, you kind of can't tell that there's any phytoplankton there. It just looks like seawater. But if we zoom in and look under a microscope, you're going to see something like this, where there's a large diversity of these phytoplankton cells. So there's a lot of different shapes, a lot of different sizes, and these cells are also doing different things, which we're going to talk about later on.
Mm-hmm.
And so I think it's important to understand that there's this hidden microscopic world there. And these phytoplankton too have different, they prefer different temperatures. So some like warmer waters, some like colder waters. Um, but yeah, I think if you were to just look at water, you couldn't really grasp that that's actually there.
Mm-hmm.
Um,
So what drives their populations?
Yeah, so number one, so phytoplankton really love to be at the surface where there's sunlight. So they're doing this photosynthesis, so they need light. And all these plankton cells, or most of these plankton cells here, have what are called chloroplasts. So that is the pigment where photosynthesis takes place. It's the green pigment, sort of like land plants also. So they need to have sunlight. They also need to have nutrients. So like land plants need fertilizers, phytoplankton require nutrients for their own growth, and largely nitrogen, phosphorus. And so those are kind of the main drivers. Also temperature. Some cells, like I mentioned, have temperature preferences. Um, but one of the things we can look at to kind of look at their
Right.
global distributions is look at a larger map. So I'm going to exit out of my slide here. and open up this phytoplankton map. So just to kind of set up this map, this is showing you concentrations of phytoplankton at the surface measured from space. So these are NASA satellites that are measuring the concentration of phytoplankton, and in particular chlorophyll, because basically we have to use chlorophyll as a proxy for how much phytoplankton is there. So for this map, in areas where you see dark green, that means there's more phytoplankton there. And blue means there's not as much phytoplankton there. And this is a time lapse. So NASA takes these satellite images every month, and I'm going to show you this time lapse. They've been doing this. This is from July 2002, and it's all the way to present day. So you can really see how phytoplankton change. But some things to point out about their global distribution: phytoplankton really love being in these coastline environments. So along the coasts of North America, South America, and Africa, phytoplankton seem to be very happy there. And one of the major reasons is because you have wind that is upwelling and bringing colder and nutrient-rich waters to the surface within those shelves, and phytoplankton grow well there. You also see a lot of phytoplankton up in the polar Arctic Ocean. That's because right now, this is in July, so there's no more ice there, right? And so there's more sunlight, nutrients, phytoplankton grow. And you're going to see as this plays in the Antarctic, right now it's covered in ice because our summer is their winter. But once it's winter time here, you'll see the ice retreat and phytoplankton bloom there also. So I'm just going to let this play so you can kind of see how plankton populations change on a global scale. And one of the things that's striking to me is just this map kind of shows you that the oceans have a real pulse to them, right? Phytoplankton populations, grow and decline, and this is due to changes in seasonality, sunlight, nutrients, temperature, ocean circulation. All these things are leading to what you're seeing here. One thing to note too, in the center of these major ocean basins, it's really blue, right? And that's because the farther away you get from land, there's less nutrients to supply those phytoplankton. And so there's going to be less of them and typically smaller cells. And so, yeah, we're almost up to 2020 now, but this is kind of a more zoomed out view. And I think we're going to zoom in also, but.
Yeah. So one of the slides you showed us right before this had kind of a various different phytoplankton in it. Can you kind of break down what types of phytoplankton there are, some examples,
Yep.
and then maybe what is most common off the coast of Georgia and why that might be, that sort of thing?
Yeah. And I think the best way to show you guys phytoplankton is to actually look at a video of them, because it's hard to kind of grasp what's really happening here. And so there's a science group out in France, actually, that made these really awesome videos. So you're going to see microscope live views, so cells that are actually live within the that they captured with a microscope. So they're going to swim around, but also some still photos too, just to show you the breadth of phytoplankton diversity. So I'll play this video and then I'll pause it a few times, but it's just a great way to kind of showcase different sizes, shapes of these different phytoplankton cells. So you can see there's really, some of them have these different spines, different external features. Some are really circular, others look more like this, which are not. This kind of still right here is a lot of groups that we see off the coast of Georgia. So these are common types of phytoplankton. And you'll notice it's not just one type, there's multiple, they're interacting with each other. And so as we're going to touch on, this can lead to some different things. But they're definitely not by themselves. There's many different kinds. Some have flagella, this little whip-like structure right here. And just to pause on this phytoplankton because I think it's fascinating. So as I mentioned at the top, phytoplankton do photosynthesis. So they are using sunlight to fix energy. But there's some types like this one here that can both do photosynthesis and also consume other cells. And we call that mixotrophy because they can mix both of these things. And you might wonder why they would do that. Well, if it's winter time and it's really cloudy and these cells are deeper in the water column and there's less sunlight, they may want to switch to ingesting cells that might be there. Vice versa, if it's a nice sunny day, if it's summer, they're up at the surface, they could switch to photosynthesis. So having the option to do both of these strategies are going to help these phytoplankton cells. They also grow these finger-like things, so they're normally not there, but they grow them out to increase the surface area of sunlight, which is just crazy. So there's a lot of different strategies these phytoplankton use.
What's the point of it moving so much? Do you know?
Yeah, so the flagella part.
Yeah.
I don't know if it's trying to keep its position in the water column.
Okay.
But the other thing is that these cells, when I say they can move, they can't move that much. So to them, when they move, it's kind of like a viscous soup kind of. So they're not really going anywhere fast. But it can help them if there's like a prey item there or some other kind of nutrient they can swim towards. So it's on like a micro scale.
Mm-hmm.
And so yeah, just highlighting just some of these awesome shapes, sizes. Chloroplast here, it's in the center. And all these groups we would see off the coast of Georgia certainly, and within the estuary also. Yeah, and I think one of the things that I think about when I see this is what are they doing,
Mm-hmm.
And I think that's what is still a real open question,
Right.
Yeah, so we're almost done here. These are just some still photos to show you like up close and personal what their cells look like, all these different beautiful structures. And some of them look really strange. And that's the last one, I think.
So I wanted to ask you about where phytoplankton fit in the food chain, right? I
Yeah.
assume they're at the base of the food chain. So maybe another way of putting it is, what is the significance of phytoplankton? How do they, bolster life
Yeah.
in the ocean?
That's a good question. So in addition to helping us breathe,
Right.
they're the base of the marine and freshwater food web. So everything that's in the ocean, either directly or indirectly, relies on phytoplankton. So you have different organisms in the water column, like shellfish that are going to filter feed phytoplankton, right? You have whales that can filter plankton. But then you also have a food chain where you have smaller grazers that are consuming these phytoplankton. The smaller grazers are ingested by larger ones. They're ingested by smaller fish, larger fish. So whether it's directly by these larger organisms or through the classic food chain, phytoplankton are the real base of all life, really.
Right. So I imagine that includes zooplankton, fish,
Yep.
pretty much anything. Yeah. So I know that they can also be infected, right? Parasites can infect them. Can you talk a little
Yeah.
bit more about that? And does that have a purpose?
Yeah, it does. So I think just to take a step back, so just to
Yeah.
kind of zoom in more on phytoplankton mortality, which is a lot of work that I focus on. So we've talked a lot about how phytoplankton grow, so nutrients, sunlight, temperature, but how do they die? So it's a
Right.
little bit morbid, but there's two main kind of ways that phytoplankton die. And one of them is grazing by tiny microscopic animals that we call microzooplankton. We can call them micrograzers, but they're essentially the same size range or a little bit larger than phytoplankton. and they consume phytoplankton really fast, almost as much as it is produced. So they can really control these phytoplankton populations. We can look at what those grazers look like. If I can figure out how to get to the next slide. So here in the left panel, here in the top left, we have a typical micrograzer. So that's what it looks like. It has a flagella, so it can swim to find prey. This was actually from a study that I did almost 10 years ago during my master's. But essentially, we have grazers that do not have any prey. When we feed them phytoplankton prey, they can ingest really fast within 20 minutes. So these green circles within the grazer are those prey cells. Within 45 minutes, we found grazers that had 4 to 6 prey items within their bodies. And after an hour, you can see some of the grazers completely stuffed themselves with this phytoplankton prey. We measured up to 10 to 12 cells per micrograzer. So the most common form is directly engulfing prey. But there's this other form of grazing that's a little bit stranger. That's in the top right here. So here we have a microzooplankton grazer. And what it does is it finds a prey item, which in this case is highlighted here, and it can extrude this feeding pouch type thing. And this pouch then surrounds this prey item. It pumps in digestive enzymes into this pouch that can break down this prey, and then it absorbs that food back up. So all of this is happening externally, which is crazy. And it happens in the matter of minutes to hours. And then on the bottom right, this is the last kind of example that shows you that even though these grazers are really small, they can consume prey items that are much bigger than their own body size. So here we have a small micrograzer, a large circular prey item, and this is what you get when you feed that grazer that circular prey item. So they might not prefer that kind of prey because it doesn't seem optimal to digest something like that. But if there's no other prey items and you give them this, they will definitely eat it. And so yeah, this is one kind of mechanism of phytoplankton loss, which is grazing.
Mm-hmm.
The other, which you mentioned, is parasite lysis, which we can talk about next.
Yeah.
And so this is where things get a little crazier. So with parasite lysis, just like humans, animals get parasites, viruses, phytoplankton also do, but it's happening on a microscopic scale. So here we have some microscope slides. They are now stained with a fluorescent dye. So in all of these panels, the parasite is in green and the host is in red. So we're just going to briefly walk through this. So in panel A here, these are little tiny parasite spores. You can think of this like land plants that will produce spores, right? But these are in the ocean, so they can swim. And so these are parasite spores swimming, trying to find their next host. Panel B is an unsuspecting host cell. And then panel C through E, that's where the parasite, which is in green, has found that host. It is infecting it and growing within inside of that host cell. And what's really fascinating about this process, about parasitism, is that the host cell can still do what it normally does. It can do photosynthesis, it can move around, it can behave normally, even though there is a parasite that is consuming it from the inside out. And so researchers have kind of likened this to a zombie-like process, where they kind of hijack their host cell. But the parasite, after 2 to 3 days, it grows so large that the host cell bursts open, which is what you get in panel F. So that's what's left of the host in the bottom right. And out comes this worm-like structure that is just wild. And so each dot within this structure is an individual spore. And so this stage only lasts for 2 to 3 minutes. It's really brief. And then the worm fractures, and we go back to step A, where you have just a bunch of spores. So you go from one spore to hundreds. And I've only captured that worm in my own microscope samples like once or twice, because it just happens so fast.
Does this happen more often in certain regions than others? Do you know?
So it's still unclear like what kind of environmental factors would drive this, but parasites love a lot of hosts. So Like we're going to talk about later, when there's a bloom, when there's a lot of plankton, parasites love that. We think it's because it's an encounter problem. So if there's more hosts there, they're going to bump into them and going to find them more often. So they really do well in coastal environments where there's a lot of phytoplankton.
Right. Okay.
Yeah.
I mean, when I think of maybe one of the benefits of it, I think of harmful algal blooms and,
Yeah.
the risks that those pose. So maybe we'll just start there. Can
Yeah.
you talk a little bit about harmful algal blooms,
Yeah.
where they occur and what risks they pose towards animals, marine life, and also humans, if they do?
Yeah.
Yeah.
That's a big question.
Okay.
We
Okay.
can,
Yeah.
we can kind of break it down. So, I guess to start, right, phytoplankton, as we talked about, love nutrients, sunlight. When you get a lot of those, when you get a lot of nutrients and the right kind of sunlight and temperature, you can get a phytoplankton bloom. So phytoplankton blooms are natural. Most are normal, right? The issue is when the species that blooms also produces toxins.
Okay.
That's what makes it a harmful algal bloom or a HAB, because they're naturally producing toxins. And so when they get really concentrated, there's going to be more toxins there. Um, and so basically, one of the major drivers of these coastal harmful blooms is runoff of nutrients. So that's fertilizers, sewer water, those kind of things that are dumping nutrients into our coastal waterways. That's going to drive phytoplankton to kind of go up. And if there's a harmful species there, then they can bloom. Um, and one might think if there's more plankton, there's more oxygen. Great, right? But not necessarily, because when you have a lot of plankton that are in the water, they're going to be dying. And once plankton die, bacteria swoop in and want to consume all that dead organic matter, all of that food source. And so bacteria swoop in and consume all that food, and they're using oxygen. So it drives the oxygen levels way down. And that can lead to things like fish kills, and it can be really bad. And so that doesn't have to be a harmful bloom. That could be any bloom. But
Right.
it's when you mix a really concentrated population with toxins that they naturally make, and then you get a harmful phytoplankton bloom.
Okay.
Um,
So I suppose that those can geographically happen anywhere, but
yeah.
are there certain areas where they are more common? Maybe, you know, uh, is there relevance here on the Skidaway River or the coast of Georgia in general?
Yeah, yeah. So they do tend to happen along like coastal regions, warmer water. So I have some examples for you. So this is one example of a harmful phytoplankton bloom in Chesapeake Bay. And this is caused by the phytoplankton species called Alexandrium, and they produce really nasty toxins. They also have natural pigments that are red-brown, which is why when they become so concentrated in the water, they form red tides,
Hmm.
which is what you can see here. And they can cover large areas because you have wind and mixing that can push these, phytoplankton. So they can cover large areas. So they do tend to happen within, coastal estuaries, bays, those kind of regions.
Hmm.
And definitely you would not want to jump in. That would be really bad.
Right.
um,
Yeah. Yeah.
because these kind of toxins can, if there's shellfish that are eating the phytoplankton or other animals, the toxins can build up, and that's really bad. And then obviously if we eat that shellfish, that can be bad too. So it's really harmful. One other example, it's not just marine and coastal, it's freshwater.
Hmm.
This is from Western Lake Erie in the Great Lakes. And this is caused by this guy here, microcystis, which forms colonies, and they can turn the water bright green. And they're really, really bad for this environment. And you can see how close it gets to the coastline. So this kind of highlights that not only are these blooms not so great for ecosystems and for us, but for tourism,
Right.
recreation, fisheries, local businesses. We've probably heard of the red tides that are off the coast of Florida causing beach closures. So it can be far reaching.
And I don't know if you're aware of are there any time studies that kind of show, this happened more often during these years than over these years? Is there any kind of like cyclical nature to that, or
There is some kind of
yeah,
like seasonal kind of
okay,
where it's, like in this case, it's mostly uh, spring, summer blooms that happen. So when it's warmer and when there's no ice, and there's plenty of nutrients, then you're going to get blooms. So there is this kind of seasonality to it, but not always, and that can kind of make it challenging.
mm-hmm,
Um, and we're actually doing some of this work. So you mentioned here off the coast of Georgia,
right,
where we
yeah,
do have some time series work that I can share with you. And I think it's really important to monitor these phytoplankton blooms. So we're starting to do that here. So we're actually looking for one species in particular, Akashiwu sanguinea, which you can see here. And just because we don't necessarily have these massive, red or green tides that you see elsewhere,
mm-hmm,
we have the species that can form them. And so it's important to gain baseline information on how these populations are currently and what's kind of driving them so we can see how they might change later on. So here at the University of Georgia, so we're actually in this building right over here, and the estuary is right here. So it's a short walk to the sampling dock. But in the Cohen Lab here at UGA, we have a PhD student, Mallory Mintz, who's up in the top right, and she's leading this kind of field sampling work
right,
where she goes out weekly, sometimes twice per week. And so here's some data that she collected that is showing you Akashiwu cell counts. So on the y-axis, we have counts of this organism, and we have month on the x-axis, and these are the years that she's been sampling. So you can see that they tend to peak in summertime,
mm-hmm,
August to October, when there's warmer temperatures. Um, and these red dots mean that they can get really concentrated, so where they could form a phytoplankton bloom. But in 2025, they didn't reach that. So currently here, I think this is real time for 2026, and we're fast approaching that kind of sampling window. So her work is really trying to figure out, is it temperature, nutrients? Are there some other factors there? Because again, there's shellfish hatcheries and there's important resources here. So we just want to make sure that we kind of know what's happening with these cells.
yeah. Can you talk a little bit more about the work that you do, and in the Cohen Lab in general?
Yep.
Speaking a little bit about the instruments that you use and kind of the techniques that you use.
Sure.
Yeah.
Yeah, so in the Cohen Lab, we do a lot of different stuff. But for me specifically, so some of the things that I do is I go out into the field and I collect water. I do microscopy, so ways that we can look at cells, and then DNA-based tools to better characterize these different phytoplankton groups. And so one of the core projects that I'm looking at is trying to characterize phytoplankton along the continental shelf ecosystems that include the northern Gulf of Mexico and off the coast of Georgia here in the South Atlantic Bight. These are really important ecosystems for fisheries and for food webs, but we don't know much about the phytoplankton there. And so really we're zooming in on these two habitats, and these are the different, these orange dots are the different field sites. So to get phytoplankton samples from these sites, you need to go out on an oceanographic cruise and gather them. So that's kind of the first step that we do. So this is from a prior cruise that I went on in the Gulf of Mexico, but it highlights some of the sampling techniques that we use in our lab. So essentially, we're on these cruises and we need to get a water sample to look at phytoplankton. So we use this instrument here called a CTD, which essentially measures properties that are in the water. So temperature, salinity, oxygen, but it also has these bottles here. And so what we do is we leave the bottles open and we drop this into the water and it sinks down, and we can tell it when to close those bottles. So essentially we're capturing phytoplankton at those depths. And then that CTD instrument comes up to the surface, we grab water from it, and then we filter our samples right there on the ship to basically concentrate phytoplankton. So that's what I'm doing on the right here, is filtering water, and we're basically concentrating it on a little small filter. And you can see there's a lot of phytoplankton there. It is very, very brown. So that's really the only way that we can capture a lot of phytoplankton in a small surface area. So that's kind of the field component. So a lot of work for just a little filter. But then we take that filter to our lab and we do a bunch of other techniques, which I won't go into too much detail here. But essentially, we want to apply DNA-based techniques to get at all the different or as many phytoplankton species as we can that are in that sample. And not only who's there, but what they're doing, who they're interacting with. And so we do a lot of different techniques in our lab for that, which the first step is basically getting all the stuff off of that filter and then doing a lot of lab work and DNA-based tools. But it's not just DNA. We also do more technical and kind of classical kind of measurements like the flow cam, which is essentially a microscope. It's an automated microscope that takes pictures of phytoplankton cells. And so that, and some examples are shown on the bottom here. So when you merge these two techniques, you get the flow cam, which can capture some of the bigger cells, and you get DNA-based tools that can capture some of the really small ones that would not show up here on this flow cam. So finding ways to, kind of holistically kind of characterize phytoplankton.
Okay.
Yeah.
I'm going to ask you a little bit more about the research too, but
Yep.
backing up and going, I had one more question about harmful algal blooms.
Mhm.
So I know that there are some human components to it, like fertilizer and that sort of thing. Is there anything that is advised that we can do to try to play our part and not adding to those? Is it just fertilizer, or is there something else that is kind of like more hidden that we might not know or be aware of?
Yeah, that's a great question. I think in terms of mitigation and what we can do, fertilizer is a big one. So I think using less of it or being more careful. There's also fertilizers now where there's lower phosphorus, so you can get better fertilizer. That might be helpful. Also, in terms of making sure your wastewater septic systems are up to date and are not leaking, that can cause a major problem. There's also buffers, so vegetation buffers that we can put to kind of separate our yards from these waterways, so that if these nutrients run off our lawns, they're going to be taken up by those buffers, plants, bushes, things like that, prior to making it in. So ways that we can kind of remove and filter nutrients before they get to these waterways. But it is difficult because also it's hard to know like where these harmful species are going to be, and that kind of changes too.
Right.
So
Yeah. Okay. So back to research, looking ahead, this can be in the Cohen Lab, your work. It can be, just research in general. Where is it headed? What are some of the biggest unanswered questions related to phytoplankton right now?
yeah, there's a lot of them. That's a
Okay.
that's a very
Sure.
loaded
Well,
question.
you can choose one or two things.
Yeah, I think my mind always goes to, and I have this figure here. It's a little bit busy, but it's from a really great paper from Karen and Hutchins in 2013, and they just summarized that these different parts of the food web. So on the bottom you have phytoplankton, the middle circle you have those micrograzers, and then on the top you have a copepod or a larger zooplankton that eats these micrograzers. At each step in the food web, you have all these different factors that are influencing them, right? You have the environmental factors, you have biological factors. And a lot of this is from the angle of phytoplankton. So what happens to them? I think I'm interested in particular looking at it from the mortality side. So how are these processes like grazing and parasitism going to change with climate change as the oceans get warmer, as there's more runoff? We don't really know because we don't have a great baseline into these different loss processes. And I kind of highlighted parasites at the very bottom because in these kind of conversations, they're not talked about that much.
Right.
It's still kind of a new field. And so I think there's a lot of work that needs to be done, not only on grazing, but on parasitism. And that will include going out to sea, collecting more samples, right? But also I think you need to pair that with laboratory work where you can have these plankton in a bottle and can control, factors like temperature, pH, CO2, and really how that changes grazing and parasite. So I think those are the things that I think about, not just phytoplankton growth, which is important, but how they're dying also.
Great. Well, I'll open it up to the audience. If anybody has any questions, or we can always just go watch some more of those videos too. Yes.
There's an article here about AMOC, the fact that the currents might be changing, slowing down, which will affect climate change.
Yeah.
How will slowing of the currents affect your specialty?
That is a really good question. That is an open question that I don't have an answer for you right now. But certainly, if there's changes in the ocean circulation and currents, it's going to influence the distribution of phytoplankton. But what I would say to that is it's hard to predict because even though currents are carrying phytoplankton, wherever they end up might not be the best for their growth, and they might not do well there. So I think we would need to really, I'm not even sure how you would study that because you need to trace an entire water mass and kind of get a lot of samples of the phytoplankton, which we don't have. So I think it's a good question. I think that also brings up the point of merging biology with physics, chemistry, because a lot of these studies don't consider biology and the biology doesn't consider physics. So I think merging both of those things.
Before we go to the next question, Wayne will be happy if I remember to turn these off so we can go to the ceiling mic. Okay, I think it was the right thing.
Sean, you showed a map of what happens to the overall number of phytoplankton over a calendar year.
Yeah.
And it seemed like that was repetitive and showed a pattern. Given that they're at the bottom of the food chain, can we see somewhat similar patterns with some of the larger animals that are ultimately eating things that eat phytoplankton? And can you correlate the two? And do you think it's cause and effect?
I think you can correlate the two. So certainly changes in phytoplankton and some of the larger species are going to influence the larger zooplankton and jellyfish and those kind of organisms. So definitely there is a correlation there. But what I would say to that is that the way that, so there is some kind of recurrent patterns, but we think that the oceans are getting warmer. That's going to shift phytoplankton smaller, right? So you're going to have smaller cells, and that's going to influence the zooplankton and the larger bracers. They might not be as happy because they prefer larger carbon-rich cells. So that could have an impact on the growth of that stage if there's smaller phytoplankton or less carbon there. And so we don't really have the data yet, but I think they're starting to predict what that would mean, right?
So that kind of leads into, if that seems logical and might be happening or might happen, then might make you wonder, as the ocean warms and you have changes in phytoplankton density that expand over more of the calendar year in different parts of the ocean, what do you think we might expect the effect of that to be on fish, for example, on the larger creatures?
Yeah, I think I'm not in a position to speculate too much right here, but I think it could influence fisheries, fish, right? If there's not as much phytoplankton biomass, or there's changes in their production, or there's not as much coastal. So what we think is going to happen is that, or what might be predicted to happen is there's a shift towards smaller groups in the open ocean. And so those groups are going to expand, and along the coast where you have these larger phytoplankton, those might get less. And that's the kind of phytoplankton that zooplankton and copepods love, which fish love. And so, yeah, if there's less of that, then are the zooplankton going to adjust and have to consume others? Is it going to influence fish? I think the answer is maybe. But sorry, I can't provide anything else for you. Yeah.
About how many different species, identified species of phytoplankton are there?
It's a really good question. I don't know off the top of my head, but I should.
Do you know if it'd be in the thousands or hundreds of thousands or?
Yeah, probably hundreds, thousands. Yeah, probably hundreds.
And so many of them aren't even yet discovered.
Well, I'm sure they're not. That's why qualified it with identified, with a term identified.
Yeah. Yeah, and there's a lot that we can't bring into the lab, and so that just makes it challenging for us to really know what's happening with a lot of these phytoplankton. So yeah, I think there's hundreds and many that we don't really know yet.
Hundreds of thousands, or
Yeah.
just like that.
So in your research slide, you showed that filter.
Yeah.
How are you able to determine the number there?
Yeah.
Accurately, and then there's many different types. How are you able to determine out of that number the different ratios of each separate one? Like, are there trace elements that they have, or?
Yeah, so we can do a lot of different things. So it's not just one technique. So with the DNA, we're sequencing all of the DNA that's on that filter, that is in that sample. And so with that, you're going to get sequence data back that we can then map to a database that tells us what that species is. So we can get relatively, with that method, it's not an absolute number, but it's relative this group versus this group. So that's about as good as we can get with DNA, but we can do other techniques like microscopy. There's other kind of more imaging-based techniques to count certain populations that we kind of know what their size are. But a lot of them, yeah, we can't get a quantitative cells per mil concentration of many of these groups. It's more relatively how are they changing? Yeah, I hope that answered.
Yeah.
This might be a little off base, but we're talking about the toxic blooms and red tide and the parasites, etc. Do any of those things contribute to the fleshy bacteria?
Oh, that's a good question. I think certainly some of the same conditions can lead to that, warming and things like that. I don't know as much about the flesh-eating bacteria side, but I would say, yeah, some of the same kind of problems there.
Vibrio prolificus will
Yeah,
become more potentially significant in warmer environments, like you said, but it's not, to my knowledge, not associated with tides, so it's a separate kind of issue.
Has there been any work to introduce parasites into a red tide to alleviate the problem?
I've gotten that question a lot, and you have to be careful because when you start, yeah.
Putting something into an
change
unnatural
something else.
environment.
Yep.
It's taken over like lionfish and stuff.
Yes,
Yeah.
but I have a colleague that's in Spain who's doing this in a controlled way, and they're trying. Their government has provided a lot of funding to answer that exact question, and it looks promising in a controlled setting, where if you kind of introduce these parasites, it can cause the bloom to go down. So it could be possible in a controlled setting. I don't know how you would extrapolate that to the actual ocean or
Anything else?
And has there been anything to determine how hurricanes increase stronger hurricane activity have on these populations of the phytoplanktons?
That's a really good question. I don't know if there's been a lot of research. There has been some research in the Gulf of Mexico that has shown that really big storms can influence phytoplankton and where they are within the Gulf. So that could certainly lead to different species being displaced into certain areas that they normally wouldn't be. But I don't know if there's been many other examples of that. But certainly if there's a hurricane or any kind of weather system, that's going to push phytoplankton, to like different, habitats that they might not normally be in.
Which would in turn push the things that feed on them.
Yeah.
Different.
Yep.
All right. Thank you very much, Dr.